And now it's my pleasure to welcome Dr. Pavithra Pathirathna, or also Pavi, thank you for that. And we will be talking today about the neuroscience of joy and aging.
Yeah, so good morning, everyone, and thank you very much for the invitation and having me here, especially Miss Christine Springer, that's Tanush, music teacher, and Miss Beth. How is everyone doing so far? Good? Yeah, good. So before we get into the presentation, let me give you a brief background of what I did in the past, so that will give you an idea why I'm here talking about this topic, right? You'll be like, why is she doing this, right? So I've been studying this brain chemistry since I was a PhD. So I did my PhD all the way in Michigan. So my PhD lab was interested in studying different aspects of brain chemistry. So back then, we were studying what is depression and, how do these antidepressant drugs work. And then we've been fabricating different sensors that can eventually go into our brains. And then once I started my career here at Florida Tech in 2019, so the focus was slightly changed, but and I'll take you to that point to give you a brief idea about what our current projects are. So I'm kind of like now studying the effect of environmental factors towards neurodegenerative diseases such as Parkinson's, Alzheimer's, things like that. And I'm trying to come up with another sensor which can actually give us some quantitative information about some events happen in the brain. And we are very fortunate that we got funding from NSF recently. So which is quite a pretty exciting thing. So having said that, let me try this one. Okay. So let me ask you this question. What is brain? Yes, that's what I was kind of looking for, right? And again, right, so if you look at this slide, right, I mean, usually when you have a speaker, right, you want your speaker to like talk about whatever like we put on the slide, right? So, but especially with that slide, I'm not going to talk about a single word, right? I mean, it's a lot of words, right? A lot of letters, right? The reason why I actually put this slide up here is to show you how complicated this brain is, okay? And as she said, right, this is the control center, right? I mean, technically, this is what actually governs the entire world, right? We use this little organ, right, to come up with all these ideas, right? And nowadays, right, we talk about artificial intelligence, right, having robots, right? And then some of our like really worried, are we actually going to get a world just like full of robots, right? What do you guys think? You think it's going to happen? Yes, to a certain degree, but we will never have a world completely governed by the robots. Why? It's because of this complexity, right? If you think about, right, we as engineers or scientists, right, are the ones who are actually going to like, like program these robots, right? But then for us to do it, we should know every single thing about this brain, right? And in 2023, we know very little about the brain. 99% we don't know, right? So we're not gonna reach to this point, oh yeah, like we know how brain works and then here's what you should be doing. So we don't have to worry about this artificial intelligence, robots, it will never, exceed us. So we as human will be the ones who will be still governing the world, okay? Good thing, huh? Another thing, if you look at this diagram, right, so the diagram has like different colors, right? So which kind of shows you different parts of the brain. And another thing is actually, so this little organ has designated areas which are actually kind of doing one specific thing, okay? And you might actually have heard something called, oh, your right brain, your left brain, right? Have you heard that? You think it's right? Is it true? Not really. It's one of the myths, okay? The reason is actually, if you think about it, right? So the analysis is mostly done by the left side of the brain, okay? And the math skills mostly from the left side, whereas art is done mostly by the right side. But think about like you playing a piano, right? Can you actually play the piano just by knowing your codes, you can't, right? Your brain should be able to like do this with a proper analysis, right? Proper logical thinking, right? So you need both the parts. So we all, we are like all brained. There's no such thing called the left brain or the right brain. That's a myth, okay? Having said that, now, let me show you an interesting diagram. Has anyone seen this one? Gosh, that's great. Yes. So what is a neuron then? One brain. Okay, great. It's called the neurons, right? So the neurons is the functional unit of the brain, okay? Let me ask you another question. How many neurons do we have on an average? Throw some numbers. So we actually have about 86 billions on an average inside this little thing, okay? And so all these little cells are kind of arranged in a form of a network. They're very close to each other, but not touching each other. We'll come to that point, okay? And of course, me being a scientist, right, would love to throw some scientific words, right? Although it might not be the the best thing of interest. But the reason why I'm actually going to take you through this little cell is it will help us understand how this brain chemistry works. And because if you know how these things work, and it's easy for us to control our own neurochemistry or own brain chemistry, okay? So this part right here is known as a cell body, which is kind of like the center of the action. And then we have this little protrusions, which are known as the dendrites, okay? So these dendrites are responsible for collecting signals from another neuron, okay? So these signals could be either chemical or electrical or any stimulation you would get from a receptor. Say, for example, when you smell, you get that signal and it'll go to the dendrites, okay? And then you see something beautiful, right? And your eyes will say, oh yeah, that's pretty cool. And then that's a signal that you would get, okay? And then as soon as these dendrites get this signal, what the cell body does, it creates something called an action potential, right? Little complicated, right? But we all are familiar with the electrical current, right? The thing that we would actually get out of our power plugs, right? So you can think of this action potential as a current, okay? Now the cell body generates this current. And this current is now going to go through or travel through this tail type thing called the axon, okay? And as you see, you have, I'm not sure whether my mice can be, oh, can you actually see my pointer up here? Yes, okay, great. So you see these little covers, okay? And then those are called the myelin sheath, okay? It's actually made up of a protein. What this little myelin sheath does is it actually accelerates the propagation of this signal. Instead of the signal trying to move slowly along this axon, now it kind of can jump. Okay, so it keeps like moving faster, okay? And then it reaches this end, which is known as the axon terminals, okay? Now, this end is pretty important because this is where we pass this signal to the next neuron, okay? And anything that actually kind of destroy this neurotransmission is going to kind of mess up with our brain, okay? And we'll come to that point. Now, any questions? You don't have to wait until the last moment. We can talk if you have any.
It's a very basic question, but
Yeah.
I kind of need a visual.
Right.
How did they first learn the size and shape of that with an electron microscope?
Yes.
Or?
Yes,
Yeah.
it is actually. You know much
Oh, no. I'm
Yes.
just wondering
Right,
because,
yeah,
you know,
we
it's
do.
so, I'm sure it's not visible to the naked eye.
Right. No, none of these are visible to naked eye. They first started with the electron microscopes. We have some of the spectroscopic techniques available now. Even much sophisticated optical microscope can still visualize. I mean, the latest ones, yes. That's a great question. Any other questions? No? Is everything?
On mine, if the pathway in between the one neuron and the neighboring neuron is that whole business called the synapse?
Yes, I'm going to take you to the next slide. You're going to
Okay.
see the whole synapse. That's a great question. 1 second, we'll be in that slide. Any other questions? No? Okay. So right here is the synapse, okay? Now, this process, as I said, is known as the neurotransmission, right? I mean, if you can just like dissect this word, right? It's neurotransmission, right? Neuro stands for the neuron, and the transmission is like the passing of this action potential or the current from dendrites or the cell body to all the way down to the axon terminal, okay? Now, if you wanted to look at this diagram up here, so this first one you see is known as the presynaptic neuron, okay? And that's an axon terminal that I just explained you, the end of an axon terminal. And this one up down here is known as a postsynaptic neuron. That's a terminal of the dendrites of the next neuron. Remember, like we said, the dendrites are the ones actually who receives the signal, and the axon is the one who actually passes the signal, okay? Now, this, you see there is a gap, right here. So this gap is about 40 to 50 nanometers, and it is the gap that I didn't actually see her name, but whoever our Zoom oh, Eugenie, okay. Yeah, was asking about this gap is known as the synapse or the synaptic cleft. And it's about 40 to 50 nanometers. You could imagine like how tiny, teeny these gaps are, right? Now the question is, now let's say we get our this signal all the way down here to presynaptic neuron, right? Now we have a gap, but we still be able to like send that signal to the postsynaptic neuron, right? How do we do that? We do it through this little chemicals known as the neurotransmitters, okay? And these are small molecules, okay? Up to now, we've found about more than 100 different neurotransmitters, and each one has specific action. And here's the thing. Now, when the action potential comes all the way down to here, these neurotransmitters now are ready to be secreted from the presynaptic neuron, and then these neurotransmitters are synthesized and stored inside the presynaptic neuron. We have little vesicles, and then these chemicals are trapped there, okay? As soon as it receives the right current or the right potential, these vesicles are going to open up, okay? And then fuse with this membrane, and then starts releasing these chemicals, okay? Now, these chemicals are in this synapse, and then it slowly diffuses. I mean, we all know what is diffusion means, right? From a high concentration to a low concentration. Now, it slowly travels to the postsynaptic neuron. Now, on this membrane, we have something called the receptors, which are actually waiting to welcome these chemicals. Okay? Now, these chemicals will get into this postsynaptic neuron membrane, okay, and then say hi to these receptors and tries to sit on these receptors. So as soon as they make that connection, they pass the signal to the postsynaptic neuron, okay? Now, let me ask you this question, right? What if the postsynaptic neuron is going to continuously receive that message, right? It's going to be a problem, right? Right? Let's say like you're so happy about that, right? And then your brain feels, okay, like I'm so happy, right? But what if that happiness is going to continuously like, you know, like firing you, right? We want it to be happy, but not for like 24/7, right? You know, that's a problem, right? Right? So then we'll be like, oh yeah, right, whatever happens, right, we're still happy, you know? So we don't want that to happen, okay? We want this neurotransmission to be regulated, right? I mean, we want it to be happy when we want it to be, and then we want it to get away with that feeling, okay? So then we need to stop firing this postsynaptic membrane or the postsynaptic neuron, okay? In other words, so now whatever secreted in the synaptic cleft has to be cleared away after the signal is transferred, okay? Now, there are different mechanisms that happens in the synaptic cleft. The easiest way would be these chemicals will slowly diffuse away from the synaptic cleft, okay? When they actually diffuses away, so there are some other cells called the we don't want to like put the names out. It's called the glial cells or the glial cells. They'll eat those neurotransmitters and remove it. Another way is there are some receptors on the presynaptic neurons that can actually reuptake those secreted chemicals, okay? That's another way. Another way is some enzymes, if you've heard about this word, right? Another type of protein, right? Those can actually come and break down all these chemicals so that the activity will stop, okay? Now, so the ones who are addicted to all these narcotic drugs, right? The problem is when someone is actually getting those drugs, it actually continuously releases these chemicals, okay? So then they live in this fantasy world, right? Every 24/7, they're happy, right? Because it won't stop this. Like the moment that the brain see, okay, like my level goes down, this drug actually gives an extra boost of those neurotransmitters. Yes, ma'am.
Is that dopamine?
Yes, I'm gonna show you that name. Great, I have a great audience, right? This is cool, really cool. So she already put up the name, right? Some of the important neurotransmitters we have. One is dopamine, okay? Another one, serotonin, right? You might actually have heard about these words, right? Another one, oxytocin, right? So these three are considered the happy neurotransmitters, which makes you feel happy, okay? As long as we have the right amount for these three in the synaptic cleft, we are good. We feel happy, okay? But we don't want too much either, but of course we don't want low levels of these either, okay? So I have this little emoji, okay, to show you. Now, on the other side, we have this one, cortisol, which is not so good, right? If the cortisol level is high in the synaptic cleft, we feel a little sad, okay? But it's okay for us to have it. As I said, we don't want it to be happy all the time, right? I mean, when we want it to be worried, yeah, why not, right? So the bottom line is we need to actually regulate these concentrations. So anything that messes up with this regulation will give us problem, okay? Any questions? I have like so many chats here, actually. So now this is how this entire neurotransmission process works, okay? And then now, because we want to know what's happened, during the aging, right? We'll come back to this neurotransmission and we'll talk about like how to regulate this properly and all these things. But now aging. Now, what will happen is, so the neurons will do their job for a good amount of time, right? At some point, everything, just like us, like the neurons will get tired, right? It'll decide, well, I'm tired, now I need a break. And that's when this aging starts kicking in, okay? And again, it's something that we cannot control. We can control, but we cannot completely stop, right? So, very natural phenomena, okay? There's nothing to be worried because some people are like I'm in my like mid-50s, right? I have like, like 10 years left, like, 10 or 20 years left. So, it's no such thing, right? I mean, it's what it is, right? There are things that we can control and there are things that we cannot control, right? So aging is one of those things. And the research has found that, as we grow with the age, the activity of the neurons decreases, right? At about 90 years, 11% of the brain mass is lost, okay? And then this mass, it's pretty significant, 11%, right? And this primarily occurs in the cerebral cortex. That's the outer coverage of the brain. That's what as soon as like you open up the skull, that's what you see. And this, especially the prefrontal area, like up here, is what actually does the planning, decision-making, cognitive decisions, memory, So then you'll start to see those things are getting affected. Okay, now the question is, right, but just because you're having trouble with these activities doesn't mean that you're aged, right? Or doesn't mean that you're actually having dementia. Okay, because, to be honest with you, right, I can't remember half of the stuff that I used to remember when I was a PhD student, right? You can't, right? So it's okay to like not to remember everything, right? I mean, I always say this because people are like, I'm losing my memory, like I don't, that's fine, right? I mean, that's normal, okay? And in the next slide, I'm gonna tell you some tips how to kind of like rewire our brain. We can, but again, it's a very natural phenomenon, okay? Any questions here? No? Good? So have you heard this term neuroplasticity, right? So this is one of the fascinating phenomena that the brain actually has. Some people think this is relatively a new idea. This is not. This was actually found early 1900s, but on records, this came around 1948. That's when like scientists actually started to like, investigate what is this neuroplasticity. So it's a relatively a new topic, but not so new. So if you dissect this word, neuro is again neurons, right, the brain cells. Plasticity is the ability to change, right? And if you wanted to read about this, you'll see like so many different definitions, but in simple, it's the ability to adapt, change, and learn, okay? Now, what do I mean by that? Now, let's say you watch a new movie. Okay, and if that is your first time watching that movie, your brain starts a connection right away about that movie, okay? And if you feel good about that movie, and then brain makes another connection saying, oh, like that's so great, okay? So you get this rewarding feeling, okay? But when you repeat the same activity, you can actually reinforce your brain to make a very strong connection. Okay, this is why, especially when we were students, we were told, right, do not wait until the last moment to study, right? If you wait until the 15th week to get your stuff done, you're not going to get it done, right? Rather, we would always say, okay, you study today and repeat it tomorrow before you start the new things, right? Because you're telling your brain, you need to somehow keep this in your memory. Like, you need to make this connection, okay? And just like we exercise to keep our, physical well-beingness, right? We can exercise our brain so that it will increase the ability to keep the stuff in the brain. Like it'll strengthen your memory, emotional regulation skills. So as a chemist, I would love to have some numbers, to say like, oh, like when you meditate, like this is what happens in the brain, right? And when you exercise, this is what happens in the brain, right? But unfortunately, such information is still not existing. But what we know is the studies have already proven you train your brain, you can actually enhance this neuroplasticity, and you can kind of delay you getting age, right? And then you can kind of control or manipulate your brain. Any questions? No? Great. Now, the important stuff, right? How do we control neurochemistry, right? So this is my least favorite thing, but this is what people have been doing the most of the time, drugs, right? You'll be like, well, you said like you're a chemist, you're a scientist, you don't like drugs? Not necessarily, right? I don't like drugs. The reason is actually, again, before I get into more details, like let's say like if you have someone like severely depressed, okay? No option, he or she has to be on antidepressant drugs if the depression is severe at that stage, okay? We can mess around at that point. We need to somehow get your serotonin level up to the right concentration. But let's say like you feel like you're stressed, okay? You're not like severely depressed, like, but you feel like, oh, it's bothering me, like I worry about this and that. At that stage, you don't have to be on antidepressant drugs, okay? You can totally get your serotonin boost up by your life changes, which I will be talking in next slide, okay? Now, why do I not like these drugs? As you see, right, this could lead to all these problems. Tolerance, dependence, withdrawal symptoms, right? Because with antidepressant drugs, these are known as the SSRIs, right? If you've heard about this, right? So what this SSRIs stands for is the specific serotonin reuptake inhibitors. Remember I said we wanted to have a high concentration or a high level of serotonin in that gap if you wanted to be happy, right? So the moment we get the levels of serotonin down, we can't be happy. Now, what these drugs do is they actually go and bind with some of the receptors, and by blocking those receptors, they kind of slows down the reuptake of serotonin. Remember I said like the one of the ways that the synapse is actually clearing these chemicals is through reuptake. So then these drugs will go and bind with those drugs. Now, here's the question that I have and then that I'm trying to actually answer through my current research is, now we know serotonin affects our mood, right? But what we don't know is when the serotonin level goes down, what happens to the level of dopamine? We don't know. What happens to the level of oxytocin? We don't know. What happens to the level of acetylcholine? We don't know. Why we don't know? Because we don't have a tool to detect those quantitative changes. Like we need something that can eventually get into our brain, right, and be able to measure these things. But then that device has to be super fast. Think about like how fast we change our feelings, right? Like in 1 nanosecond, right? We can go to like Europe, right, and then back to Mass, right, and then back to Florida, right, in like few seconds, right? So then whatever our tool should be able to capture those ultra-fast tools. We don't have such tools. What we have is we have a tool that can detect one at a time. Like we can say, oh yeah, here's a serotonin concentration, here's a dopamine concentration. In fact, that's what I used in my PhD lab. So my lab is like more focused on serotonin. And these are actually Chinese sensors, which are made up of carbon fibers, and the diameter is about 5 micron. Of course, we cannot get into like one single synapse, but we would probe like 100 synapses and tell, oh, like here's the concentration of serotonin, okay? That's the information that are available for these pharmaceutical companies. So then they target one thing and then give us the drug, okay? And if you do a small Google search and see like how much has been invested on this neurochemistry related research versus the curing rate, there's no relationship, okay? So the research has been like going up and up and up, right? But the curing, there's no change, right? It's because we don't have the right information, right? I still remember like during the PhD, so we did our drugs. Most of the SSRIs we administered those drugs to these mice, the depressed mice models, and we tested the changes in the serotonin. We didn't really see much of a decrease, okay? So things are not quite right, and it becomes even complicated because from one to another, I mean, the brain is different, right? So the way that your brain works is going to be different. I know. Yes. Yes.
Doctor, I became aware of a DNA test recently. I think it's called GenSight. Mm-hmm. And so a person sends in their DNA, and then that's analyzed to pair that person with the most effective antidepressant based on their DNA. What are your thoughts on that? Is that?
That's a great question. So right now, actually, we are moving towards this genetic-based analysis. It's because, say, for example, I'm going to come back to your question now. If you've read about most of the cancer-related research, right? And then for years, we worried these cancers are actually caused by some external factors, right? But now we realized most of the cancers are actually coming from our genes, like it's genetic-based. That's why we cannot actually find a one universal cure for cancers, right? So the same thing goes with these things too. And then one aspect of this neurochemistry, or like how we control these things, is there is a genetic base. So what it does is actually compare your genetic code versus, because with the standards, because the standards were done, okay, like if this person has this particular gene, this drug might work. So we have the standards, and then they would compare your genetic code with the standards that they have and give that drug to you. That's what it works. It does work. I'm not, especially, say for example, for dementia, Alzheimer, the neurodegenerative diseases things, they work. But the question is like, is it 100%? Not yet. So, my suggestion is like, especially if you have young folks, right, who thinks that they're depressed and they need to be on antidepressant drugs, I would not, put them on the drugs first. Rather, I'll try these methods. No, that's a great comment, because it's always recommended when you are even on drugs, as you said, you need to actually be paired it with the lifestyle changes, like some sort of a positive reinforcement. So that way you can easily get away from the drugs, because all what you need is this boost, like once the brain realizes, okay, like this is the serotonin concentration that I need to keep in her brain so that she's happy, the brain will do it. But to get that boost, people put on antidepressant drugs. I mean, as you said, if you're clinically diagnosed, no option, you have to go and get the drug.
Is it possible for many people who started with the boosting serotonin and SSRIs and the others, to get off of them one day? Or?
Oh, for sure, 100%. 100%, yeah. That's when the neuroplasticity comes in, actually. Exactly. So that's 100% with you, because my PhD lab, the stuff that we did during my PhD, so the findings that we had, we had such a hard time, selling it to the community, because all the pharmaceutical companies were like, no, what you're saying is not right. And we were shocked. We were like, no. This is what we find, but we always have that kind of like a clash, Yeah, go ahead.
Yeah, it amazes me that I find these people that have been on like Prozac or something like this for 20 years or something. I'm like,
nothing. Yeah, we tested one. That was like one of our drugs, actually. We tested. It doesn't do anything. Yeah, and all what it does, the number one thing that they do is like put you in sleep, right? So they make you sleep, right? And then under here, like as one of the lifestyle changes, I also recommend, sleep, because the more you sleep, the more you give, it's like you're giving more rest to your brain, right? And then it kind of like, and think about this, right? Like, let's say like you cry for like one whole day, right? And then all of a sudden you fall asleep, right? And then when you wake up, you don't feel that miserable, right? And you're kind of like relaxed, right? So most of these antidepressant drugs do that as their number one job, like put you on sleep, right? And then, as I said, I'm not saying like none of the drugs work, but it does work for a very small population. But the majority, it really doesn't help as compared to lifestyle changes. But if you do practice any of these things, 100% guaranteed, you can actually get rid of this depression. Because here, doing all these things, you're not like pumping, right, like tons of chemicals at once to the brain, and you're slowly telling the brain, okay, like, here's what I need, like, can you just like give me this, like just the neuroplasticity concept, like you keep reinforcing your brain saying like, this is what I want, can you just do it for me, this is what I want, just like studying, right? If you study the same material, right, like hard enough, right, you're gonna remember, right? And when people say like, oh, like you're doing chemistry, it's so hard, and I'm like, my answer is like, no, it's not hard. It's because you didn't learn that in the right way, right? But if you're given enough time, and then study it properly, and if you have a brain that functions normally, everyone should be able to like, train your brain. Just like that's what meditation does, right? So then people say like, do meditation, and the research have like proven those things work 100%. But still, what we don't know is what exactly actually changes like during those events. But if you think about this diet actually affects this in several ways. So the serotonin, the 90% of the serotonin is produced in the GI tract, not in the brain, okay? So, and then if you think about the number of neuronal connection our GI tract has, it's millions, okay? So then there is this gut-brain connection that people have been studying a lot recently. And it says, like, say, for example, depression can be detected at an early stage because one of the complaints that people have is like they start to like feel like tummy pains. Okay, one of the early symptoms, yeah. So now people are kind of like moving, can we actually detect this at an early stage so that these therapies can be started? Because if you wait until it affects this entire system, we might be too late. Another thing this diet can do to us is most of these neurotransmitters are proteins, so we need good proteins. If you give yourself like good proteins, so then it actually kind of like boosts the concentration of these neurotransmitters, your body can reproduce, keep generating this good stuff, okay? Another thing, if you've heard about this word oxidative stress, have you heard this word, right? So this is actually, I mean, the body can sometimes generate some waste, right? This oxygen radical. So these are not good things, right? What they do is they actually come and like kind of like eat your neuron cells. So we don't want that. Now, when you actually take the food rich with antioxidants, fresh vegetables, fresh fruits, right, the fresh stuff, they can actually go fight with these oxygen radicals and kind of like stop or prevent you from getting this oxidative stress. So the diet can affect your mental health a lot. But then we don't want to eat too much sugar. Too much sugar is not good. We don't want to like eat too much, like, any sort of like carbs, right? When I said the carbs, I'm more focusing on the sugar stuff. Again, how these specific concentrations are changing, not yet known, but it will. Okay, that's one of the growing research areas right now. Of course, exercise. Tons of studies have proven it will actually accelerate your mental health. It just like give you this feeling, right? Joyness, right? Happiness, like you feel like so relaxed. So then you keep doing it, right? And then eventually your brain will come to a point, okay, like I cannot make her, worried because this is what she wants. Like she wants to be happy. And then it'll do it. Okay, and social interactions, just like what we do now, right? And the bottom line is you wanted to think about only the positive side of everything, right? I always say, like, even if I feel like, like someone passed away, I think that's the best thing that could have happened at that point, right? Take it as, everything is the positive, yeah, just like everything happens for the best, right? I mean, yeah, we worry, but say, for example, if that person is in no pain any longer, right? I mean, it worries us, but at the same time, it's good for the person who passed away. I mean, like we should take it like that, right? I mean, but the more we worry, right, and the brain thinks, oh, like this is like her thing, and then it's gonna like actually lower the serotonin concentration, lower the dopamine, right, and then put more cortisol. We don't want to do that. So this is why this good things, the positive vibes. Helps neuroplasticity. And there's another concept which I didn't include here. It's called the neurogenesis. That's the ability of brain to grow, okay? So in the past, people thought once you're born with like this many neurons, that's it. That's not true anymore. And studies have proved that even in adults, in some cases, the brain can grow. Okay, and it's again like one of the growing research areas in the field too, to see like how we can actually kind of use this neurogenesis together with neuroplasticity to like come up, but to find better solutions to move away from all these neurodegenerative diseases as well as some depression-related stuff. Okay, so with that being said, now this is some of research work, just to give you an idea about actually where we stand as a neurochemist, neuroscientist. So this question of the serotonin depletion always bothers me. I mean, still I don't believe that's just the serotonin does this depression. Because we don't have any evidence of any other neurotransmitters. So then that's telling me like you can, just because we don't have enough information doesn't mean that you can say like, oh, there's no effect from dopamine, there's no effect from ascorbic acid, there's no effect from GABA, right? We can't come to that conclusion, okay? Now, what I'm trying with my current research group up here is like all my PhDs and master's students and few undergrads. My lab is pretty large, actually, but this is like half of the lab. So what we are trying to do is to come up with this little sensor where we can actually kind of like functionalize each of these sensors. You can see that this one has 4 sensors, right? And then one will give us information about dopamine, while the other one gives information about the serotonin, the other one maybe like ascorbic acid. Now, I put this as a copper, or like I put it copper as one of the toxic metals that we have, because one of the other things that I wanted to actually understand is the effect of environmental substances, such as heavy metals or pesticides, on neurodegenerative diseases, say, for example, Parkinson's, like Alzheimer's, because not like, if you look at the population who are affected with the Parkinson's, they're not like 70 plus people. I mean, you have like very young people actually who are getting this Parkinson's. And if you do a little background search, most of these people are somehow exposing to some sort of a heavy metal containing thing, right? Like an agricultural field, like pesticides, insecticides, you know, they've been like touching this for a long time. And then they end up getting this Parkinson's at a very early stage, like 50s. 50s is not an age for you to have Parkinson's. It's not normal. But again, right, we were interested in understanding how aging actually caused all these problems, but we never paid attention to this stuff, right? And so then that's another thing that I wanted to do. So then let's say like copper, chromium, we do like a lot of research, chromium, arsenic, lead, and we are fabricating different sensors to detect all those metal ions in our body too, like eventually get into the kidney and stuff. So hopefully, this will eventually get into the mice brain. We're done with all the in vitro part, like the beaker stuff. We've actually optimized everything. And each of these electrode is about 5 micron diameter, and the whole thing is about like 0.1 millimeter. So it's very tiny. You can dig tiny holes and put it in the brain and measure those. So we can actually, so we would take the mice and we would give him some anesthesia and like lie him down, and then you can actually drill teeny holes. And we know we have like the maps of the different regions of the brain. Say, for example, like if I wanted to go to like prefrontal or back of the thing, we can actually use that map to find where to dig the hole. And then you would insert that electrode carefully, and you can measure it. And it is actually. So this is the fastest way to measure these neurotransmission events. And I can actually generate a signal every 100 milliseconds. This is the fastest we have so far in the world. This is what we got funding. Pretty exciting, but hopefully, like in the long run, we should be able to tell the companies, look, like it's not just serotonin. That's like my goal. I don't believe it's just the serotonin. Then they can actually try to like incorporate other other neurotransmitters in their drugs and try to somehow play with it and see what can we do. It's hard, but I will get in, because the fabricating the sensors, I mean, my kids do it using their hands. I mean, imagine cutting them under the microscope to like, to this, the length is about 150 microns, so they have to be like very, their hands should be like very precise and things like that. But we can do it. I mean, people are doing it. It's interesting, yeah. So with that, I'm going to stop here.
The role of hallucinogenics in the production of new neuronal pathways, the neuroplasticity.
Yeah.
With the treatment of depression.
Yes.
And administering these, this is what I was told, that wherever the area of trauma, that it starts to develop these neuronal pathways around that trauma to bypass it, so it's not so there in your face all the time.
Right. Yes, that's what they would predict. And then again, here's the thing. So all this, one of the challenge that they have, especially like understanding what actually does, is we cannot actually map this in a real brain. Like when the brain is like, in terms of the real-time measurements, we can't do that, right? So what we do is actually we would most likely take like a dead brain, right? We would slice it and then we do like all those imaging, right? The question again is, right, can we actually transfer that information to a living system, right? My understanding is no, I don't believe that we can, because when you're live versus when you're not live, it's two different things, right? But then this sensor is actually, you can use it live, like you can measure the real-time changes. So based on the studies that they found, yes, that's their claim, that they think it actually kind of bypasses, like makes the neuronal new connections. But again, how well they do is a question. You know, like what's the strength of that connection is a problem. That's why they would say like do it as like a repetitive thing. But then you need to be careful about the other symptoms that you would come as a secondary symptom.
Go on. They're asking online, Kristen is asking how activities in music affect the brain.
Yes, that's a great question, Christine. It's again, what research has actually found is this listening to music actually boosts your neuroplasticity. And in terms of chemical, again, like in terms of like what's actually happening, no information yet, but all what they know is like you listen to music, it actually kind of help it increases the neurogenesis, because there are studies that they've done with the people who were paralyzed, like the brain was like completely like sort of like shut down, but then you let the people to like listen to this new music and the brain slowly comes back up. So that's, at that point, it's more like a neurogenesis actually, because the brain has to like develop new cells. This is why they now say it's this neurogenesis now actually happens even in adults, not at just the younger age. Of course, it does increase the neuroplasticity, makes the strong connection. Now, as a chemist, what I would think is, as you actually establish this new connection, you again reinforcing your brain and say give that message saying, well, I need dopamine, I need serotonin, I need good chemicals, so please keep boosting that up, that connection. But how it works, no one knows actually. It's not something easy to find exactly. I mean, if you think about it, right? I mean, we should be able to like detect it as in a live human. It's going to be pretty hard.
I'll be quick. This is more of a response than a question. When I used to work at Circles of Care on the geriatric unit, and I'd bring in music. Some of the patients there that had dementia and had balance problems, as soon as I'd put on a piece of music that would trigger their memory, they would get up and do the and dance with me and be perfect on step and everything.
Yeah.
It was amazing.
It is amazing
to witness that.
It is amazing. Yeah.
Thank you so much for speaking with us today and
No
answering all
worries.
our
Thank you for
questions.
the attention. Thank you.
Very much appreciated.